Welcome to this tutorial on protein translation. Since most of you have seen this topic many times in your life, in high school and undergrad education, this topic will be covered at the basic level in this module. I included also videos that complement the figures in the handout. You can also access the videos from the printed handouts. To see instructions on how to do that, click on Resources. So if you choose to study from the handouts, you still have access to the videos. The problem of translating the nucleotide language of nucleic acids into the amino acid language of proteins was solved in the late 1950s, early 1960s by Marshall Nirenberg and his group. So what we know now is that there are the messenger RNA nucleotide triplets code for one specific amino acid. There are 64 different combinations of bases, and 61 of them code for the 20 amino acids. There are 3 codons that don't code for any amino acids, and they're called termination codons, and they stop the translation. And there is one codon, AUG, which is also called the start codon, that sets the reading frame for the transcript and always codes for methionine in eukaryotes or formyl methionine in prokaryotes. Early on, scientists recognized that because amino acids cannot bind directly to the set of 3 nucleotides that form the codons, adapters were required. These adapters are what we call now transfer RNA or tRNA. Each tRNA molecule contains an anticodon and binds covalently to a specific amino acid. The anticodon of the tRNA is a set of 3 nucleotides that can interact with the codon on the messenger RNA. To interact, the codon and anticodon must be complementary. In other words, they must be able to form base pairs in an anti-parallel orientation. And so the anticodon of a transfer RNA serves as a link between the messenger RNA codon and the amino acid that the codon specifies.
Once the DNA double helix had been discovered, the next big challenge was to work out how the 4 letters of DNA could code for each of the 20 amino acids that make protein. The first question was, how many DNA letters coded for each amino acid? If it was 1 DNA letter for 1 amino acid, then you could only code for a maximum of 4 amino acids. 2 letters in every possible combination could code for up to 16 amino acids. Still not enough. But 3 DNA letters provide more than enough combinations to code for all 20 amino acids. So 3 was the answer. It was a triplet code.
There are 4 main characteristics of the genetic code that you should be familiar with. One is that the code is degenerate in the sense that more than 1 codon specified for a particular amino acid. This doesn't mean that the code is ambiguous. Each codon only specifies for 1 amino acid. The code is also non-overlapping, so successive triplets are read in order, 3 nucleotides at a time. The third is the code is commaless or unpunctuated. That means that there are no breaks or markers to distinguish one codon from another, although some codons have extra meaning, like the start and termination codons. And lastly, the code is universal. The same codon specifies the same amino acid in all species, with some exceptions, like in the mitochondria, for example, there are 3 codons that's specified for different amino acids. In the now classic experiment by Nuremberg to determine the specific codons for each amino acid, he used a polynucleotide polyuracil in an in vitro translation system that had all the components for translation and obtained a polyalanine, indicating that 3 uracils or 3 U's was the code for alanine. Using similar approaches, they finished the code in the 1960s, and these tables show the genetic code for the two components, the DNA and the RNA. Closer inspection of the codon table shows that in most instances there are multiple codons for a single amino acid, and the variation seems to occur in the third base of the codon. F.Crick noted this and also noted that the pairing between the 3' base of the codon and the 5' base of the anticodon didn't always follow the strict rules that he and Watson had previously discovered, that is A pairs to U and G pairs to C. So this observation led him to formulate the wobble hypothesis, in which at that position the base pairs can wobble. For example, G can pair with a U, and A, C, and U can pair with an unusual base hypoxanthine found in tRNA. So 3 of the 4 codons for glycine, as we see in this figure, GCU, GGC, and GGA, can pair with one single tRNA that contains the anticodon 5' ICC 3'. Why is this important? If each of the 61 codons for amino acids required a distinct tRNA, cells will have to maintain 61 tRNAs. However, because of the wobble between the codon and anticodon, fewer than 61 tRNAs are required to translate the genetic code. So let's talk a little bit about the conventions when representing DNA sequences, messenger RNA sequences, and peptide chains. The most accepted convention is that the DNA is represented with a DNA template strand, usually below the DNA coding strand. Then after transcription, the sequence of the mRNA is complementary and antiparallel to the DNA's template strand, and thus is the same as the DNA's coding strand, except that the T's are replaced by U's. And then the peptide sequence can be expressed with the 3-letter code or the 1-letter code, as we saw in block 1. So what do we need to make proteins? The cell dedicates vast amounts of resources to the translation process. It is the most expensive from the energy point of view, and molecules involved in translation can account as much as 30% of the total cell mass. So we need ribosomes, tRNA and messenger RNA, obviously. We need an amino acid pool closely regulated. We need initiation, elongation, and termination factors. These are mostly GTP binding proteins or nucleotide binding proteins, and the nomenclature varies whether if it's prokaryote or eukaryotes. The eukaryote factors are denoted by the E in front of them. Then we need an energy source, and these are ATP and GTP. And lastly, we have the aminoacyl-tRNA synthetase that loads the amino acids on the tRNA. So the first step is the loading of the tRNA with amino acids. Amino acids are attached to their tRNA by highly specific enzymes known as aminoacyl-tRNA synthetases. There are 20 or 21 if you count the synthetase for the initial methionine, and each enzyme recognizes one particular amino acid and all the different tRNAs that carry that amino acid. The amino acid is first activated with ATP and then transferred to the tRNA. So click on the image to see a video about the molecular details of this reaction, and when you come back we'll talk about the importance of this process. This step is extremely important for the fidelity of the translation process. Erroneous coupling of the tRNA and the amino acid will lead to the wrong amino acid being incorporated into the polypeptide, since the discrimination of the amino acid during the protein synthesis comes only from the recognition of the anticodon of a tRNA by a codon on the messenger RNA, and not by the recognition of the amino acid. So these enzymes usually check their work, and if the incorrect amino acid has been linked to a particular tRNA, the enzyme will remove the amino acid from the tRNA and try again using the correct amino acid. Of the ribosome, we're only going to focus on 3 binding sites that participate directly on the protein synthesis mechanism. So one is the acceptor site, or A site, which binds the aminoacyl-tRNA, that is a tRNA bound to an amino acid. The P site, or peptidyl site, that binds to a tRNA with the growing polypeptide chain. And then the exit site, where the free tRNA is located before it exits the ribosome. So how do ribosomes find their initiation sites? In eukaryotes, initiation process begins at the first AUG downstream from the messenger RNA cap. Prokaryotes don't have capped messenger RNA, so there is a specific sequence called the Shine-Delgarno box that binds directly to the 30S ribosomal subunit. The initiation phase of the translation process begins when a special aminoacyl-tRNA, which is a methionyl-tRNA for the initial methionine, interacts with the initiation factor 2 and form a complex with the small ribosomal subunit. Once this happens, the messenger RNA comes and forms a ternary complex with the help of other factors, and this complex is formed at the cap of the messenger RNA. From there, the whole complex scans the messenger RNA sequence until it finds the first AUG codon. Once this happens, the large ribosomal subunit binds and the ribosome is complete. One thing to note is that the initial methionyl-tRNA is bound to the AUG start codon at the P site of the ribosome. In the next phase, the elongation, the incoming aminoacyl-tRNA, in this case we have a valine tRNA, it binds into the acceptor site, and then the activity peptidyl transferase from the ribosome will move or form the peptide bond between the methionine of the tRNA in the P site and the valine in the A site. After that, with the help of the elongation factor 2 and GTP hydrolysis, the peptidyl-tRNA along with the attached mRNA moves from the A site to the P site, and the uncharged tRNA moves to the E site before being released from the ribosome. The elongation factor 2 is very important for this step. This puts the next codon in the messenger RNA into the A site. The elongation and translocation steps are repeated until a termination codon moves into the A site.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
When this happens, and because tRNAs cannot pair with stop codons, release factors bind to the ribosome instead, and then the peptidyl transferase hydrolyzes the bond between the peptide chain and the tRNA on the P site, and therefore the peptide is released from the ribosome. The ribosome then dissociates into its subunits and can be recycled to be used again for protein synthesis. As one ribosome moves along the messenger RNA, producing a polypeptide chain, a second ribosome can bind to the vacant 5' end to the messenger RNA. Many ribosomes can simultaneously translate a single messenger RNA, forming a complex known as polysome. A single ribosome covers about 80 nucleotides of the messenger RNA. Therefore, ribosomes are positioned on the messenger RNA at intervals of approximately 100 nucleotides. The growing polypeptide chains attached to the ribosomes become longer as each ribosome moves from the 5' end towards the 3' end of the messenger RNA. Protein synthesis can be regulated or inhibited at many steps. One important step of regulation is the initiation. This is done through the initiation factors, mainly initiation factor 2 and 4. The regulation is usually done by phosphorylation, dephosphorylation of the factors or inhibitors of the factors. And for example, insulin, which is an anabolic hormone, produces a generalized increase in protein synthesis, and this is done by phosphorylating an inhibitor of initiation factor 4, and therefore the level of protein synthesis increases. In the case of initiation factor 2, there are several conditions that inhibit its function. For example, starvation, heat shock, interferon produced during viral infections, all result in phosphorylation of the initiation factor 2 by a specific kinase and therefore inhibit protein synthesis. One example of physiological regulation of protein synthesis is the regulation of globin synthesis by heme in reticulocytes. The intracellular levels of heme dictate how fast the globin protein synthesis will go, and heme does that by preventing the phosphorylation of initiation factor 2 and therefore increasing protein synthesis. In the next slide, we are going to talk about the protein synthesis inhibition by antibiotics. Many antibiotics that are used to combat bacterial infections in humans take advantage of the differences between the mechanisms for protein synthesis in prokaryotes and eukaryotes. We are also going to cover a couple of toxins that also block the protein synthesis. This is an interactive slide that shows the positions where different antibiotics inhibit protein synthesis and whether the inhibition happens in prokaryotes, eukaryotes, or both. There's also diphtheria toxin and ricin, which are toxins that also block protein synthesis. So click on the markers and study the mechanisms for all these compounds. The first post-translational modification that happens to proteins, we can say, is folding. As the proteins emerge from the ribosome, they are immediately bound by chaperones that help in that folding. The post-translational modifications can happen in the cytoplasm or in the lumen of the ER. Throughout the course, we have seen many examples of post-translational modifications. We looked at glycosylation. That could be O-link glycosylation, usually happens in serines or threonines. And we have seen also the N-link glycosylation, which happens on asparagines. N-link glycosylation happens on the ER, and we will look at this later. We have seen also lipid addition that could be by modifications by fatty acids, like palmitoylation or myristoylation, or prenylation. Lipid modification makes otherwise soluble proteins associate with membrane. And then other examples of regulation are phosphorylation and acetylation. We've seen this many times. And we recently saw ADP ribosylation as a mechanism of action for some toxins, like the diphtheria toxin. Other modified amino acids that help in folding are hydroxylation of proline, for example, or the carboxylation of amino acids, very important in clotting. Membrane proteins, or proteins that are going to be secreted or destined to certain organelles like the lysosomes, are translated in a special subset of ribosomes that are bound to the ER membrane. In this case, a signal peptide, which is a hydrophobic sequence flanked by charged amino acids, is recognized as soon as it comes out of the ribosome by a protein called the signal recognition particle, or SRP. Once SRP recognizes the signal peptide, it arrests protein translation, and the whole ribosome with the messenger RNA, or the whole polyribosome, moves to the membrane of the endoplasmic reticulum, where there is a receptor for the SRP. This receptor transfers the nascent polypeptide chain and the ribosome to a pore, a proteinaceous pore, and then the translation can resume, and the nascent polypeptide is transferred vectorially into the lumen of the ER. Once inside, the signal peptide is cleaved, and post-translational modifications of the protein occur as the protein is being translated. You will see these in much more detail in cell and tissue biology, so we're just going to cover the basics. Click on the markers to study these mechanisms. Let's talk a little bit about default destination for proteins made in the cytosol or in the ER membranes. Since protein synthesis happens in the cytosol, by default, newly synthesized proteins remain in the cytosolic compartment. Some proteins, like the ones destined for secretion or for membrane proteins or protein components of different organelles, have to be either synthesized at the organelle or have to be sorted to the different organelles. And this is done through targeting motifs or special targeting sequences that are built in the primary sequence of the protein. So, for example, proteins destined to the peroxisomes will have a C-terminal, serine, lysine, leucine tripeptide that will target those proteins to that organelle. Other examples of cytosolic proteins that are transported to different organelles are the nuclear localization signals that target the protein to the nucleus, to the nucleoplasm, and the mitochondrial targeting sequences that don't have a consensus sequence but are recognized by the mitochondria and imported by the import machinery. For proteins made in the RAF-ER, the default destination depends on the protein. Membrane proteins are usually targeted to the plasma membrane or the membranes of the secretory pathway, like that is the ER and the Golgi. For luminal proteins, the default destination is secretion, unless they have targeting sequences, like the luminal proteins that reside in the ER lumen have a C-terminal tetrapeptide, K-del is called, and that retains them in the lumen of the ER. There is a receptor that recognizes that tetrapeptide and retains those proteins in the ER. And then, for example, for proteins destined to the lysosomes, there is a post-translational modification in which mannose 6-phosphate is added to the sugar moiety of those proteins. This is later recognized and deviated from being secreted and trafficked to the lysosomes.
The signal sequence of a membrane protein directs the ribosome to the RAF-ER. The growing polypeptide chain enters the lumen of the ER through a protein import channel in the membrane. There, the signal sequence is cleaved, sugars are added, and the protein folds. Some enzymes or structural proteins remain in the ER. Most proteins that are soluble in the ER are transported in vesicles to the cis-Golgi. In the cisternal maturation model, the cis-Golgi matures to become the medial Golgi. A new cis-Golgi is formed by fusion of ER vesicles. The maturation process includes retrograde vesicular transport of resident Golgi proteins. Proteins destined for secretion after modification in the Golgi are transported from the trans-Golgi to the plasma membrane. When the membranes fuse, the proteins are released into the extracellular space. Soluble proteins on the outside of a cell can re-enter by endocytosis and are sorted to lysosomes for degradation. Other proteins, including lysosomal enzymes, sort directly to lysosomes from the trans-Golgi.